Hello, everybody. I hope you can see my screen. I'm Aravind Venkatesan. I'm a senior data scientist at the EBI. I'm part of the literature service group. And for the last 4 years, I've been working on various aspects of data integration, text mining, data mining, and text analytics. And a significant part of my work has been for the ELIXIR data platform. So today in the talk, I will be giving you an overview about firstly what ELIXIR data platform does, what role your PMC plays in that, and what are the kind of work we are doing specifically under the ELIXIR context. So it should give you an idea of what's happening. Yeah. So to start off with, what is ELIXIR data platform all about? So mainly ELIXIR data platform aims to drive the use of life science data, and also and it facilitates aspects to reuse the data. And the main aim is to provide users with a robust, long-term, sustainable data resources. And apart from the sustainability of these data resources, it's also looking at the scalability and how interconnected data resources are. It is extremely important that all the data resources are well connected. That would facilitate much better. It would sort of enhance knowledge discovery within the life sciences. And the ELIXIR data platform work can be broadly divided into 4 aspects or tasks. The first aspect would be the administration and support of what ELIXIR calls the core data resources and deposition databases. These are core resources that are extremely important for the life science domain, and it supports in way of providing metrics and so on. The other aspect is literature data integration, which is to do with the literature, that is scientists publishing papers, and how it is connected to the underlying data. And I'll talk a little bit more about it in a bit. And the other aspect is scalable curation. As you may know, that a significant part of ELIXIR data resources are maintained by expert human curators. And it's not a trivial task to maintain these resources, and these experts need help, especially the background of humongous papers being published, scientific papers being published. Certainly, there needs to be tools and services that can help them find the needed biological assertions that then they can sort of that will help them maintain the knowledge base. The fourth aspect is more. It looks at how what sort of support ELIXIR can provide to these core data resources in terms of getting the word out there, in terms of financially, basically, it looks at the financial aspects. In that sense, the task 1 and 4 is more administrative and outreach support aspect. The task 2 and 3 is more technical in nature, where we are talking about how to connect data, how to represent data, and generally make a data workflow that will assist the users, the large users that ELIXIR has. In today's talk, I will mainly concentrate on the literature data integration aspect. In that, I will give an overview of what we have done so far and how what Europe PMC, what role Europe PMC plays in this specific task, and what other collaborators of the ELIXIR data platform, how they contribute to this task. So moving ahead, it is we need to understand, in order to understand Europe PMC's role, we need to understand what Europe PMC is. So Europe PMC is essentially a free digital archive for biomedical and life science research publications. So it is a core data, ELIXIR core data resource, and it hosts a variety of scientific data, that is abstracts, Agricola records, preprints, NHS guidelines, patents, and also full text articles. It is mainly developed at the EBI and partner to the PubMed Central, and it is also funded by 29 international funders who choose Europe PMC as their primary source for maintaining the articles. What is the difference between PubMed PMC and Europe PMC? The main thing is Europe PMC how hosts both abstracts and full text articles, which means you don't need to go to two different sites for searching. Any keyword would, any query would essentially search both full text and abstracts. And in general, Europe PMC is very conscious about the complexity of paper. That is, typically a research paper is not just written text. It is a knowledge base in its own right, where you have different layers of information. You have, first of all, the scientific findings, methods, and the figures information in the paper. Apart from that, you have author information, funding information, and affiliations of the author and the other authors, and also metrics that are behind the paper in terms of citations, if there are any comments, what are the wider impact the paper has, and also the links to various databases and what sort of concepts is described in the paper. So there are various layers, and Europe PMC, the services offered by Europe PMC provides a straightforward way to access this information. And this sort of flexibility in accessing data is necessary because literature is very much a part of the big data landscape in biology. So, for instance, in Europe PMC, at least you have over 36 million biomedical abstracts, and I mean it's not a trivial task for scientists, curators, and researchers to go through this all the richness of information contained in the scientific publication and extract the key information they are looking at. It's almost like finding a needle in a haystack. And as bioinformaticians, I think we need to make this task as simpler as possible. And this is where the goal of ELIXIR comes into play. This is where there is a sort of alignment of what services Europe PMC provides and what ELIXIR wants to offer is aligned. And mainly in this way, what we want to do is, in the context of ELIXIR, is create an interconnected data ecosystem where you have databases and all the citations made on with respect to the data records being reflected in the in Europe PMC in the articles. That is one layer. The other layer is to add additional information extracted from literature, that is mainly by text and data mining, and you need an infrastructure to support all this work. So this task would also involve sort of establishing the infrastructure that would ultimately support searching, triaging for of relevant information for scientists and curators alike. So moving on, how have we implemented this literature data linking? And as I said, there is two aspects to it. One is linking the available information, which is more of you can say, it's a engineering solution where you make the corresponding connection between the data records in various databases to the literature. That is one layer. And the other layer is added knowledge by extracting information mentioned in the literature. So let's see how an example of the first one, that is how the data is connected to literature. So let's first of all, this is a typical search page in Europe PMC, and let's say you're searching for P53, the protein. So it gives you date. It gives you a list of papers that mentions P53, and as you can see on the left, you can filter your search either by if you want to see only research articles or reviews or preprints. You may choose as according to your need. And let's say you want to know all the papers that are mentioned in the INTACT database, which is also an ELIXIR core data resource. So then what do we do? So there is a advanced search function. So when you click on that, it gives you additional it gives you more functionality to more flexibility to search, to add to your search. So then you would want to click on the data links tab, where as it mentions, there are articles cited in the INTACT database. So this automatically gets added to your original search query, which would be P53. I want to know what P53, all the papers that mentions P53 and the papers that are being cited in INTACT database. So then it gives you a list of papers. So this is not all. There much more to do about more information that can be received from this. So let's see from this list. If you take a sample paper, first of all, you'll notice that on the left side, both abstract and full text articles are on the same page. So you can just scroll down and go through both sections of the paper. And you may also see that there is a data section in the tab on the left. So on clicking on the data section, you can clearly see all the databases that have cited this particular article. For example, here the gene annotation ontology has cited this article, has referenced this, and also all the specific protein-protein interaction data records that cites this article. This way, this is one layer of integration. And it's not only available on the front end, you can also programmatically access this via the Europe PMC Article API. So this API provides modules to retrieve a different sort of metadata from hosted by Europe PMC. And you can sort of, for the query I just presented, you have to just use the search functionality, and that would be of something like this. So you basically have the URL, the query, the basic URL, which gives you a quick search functionality, and you just add your the query of your interest, that is P53 and articles that has intact publications and in the JSON format, and then it lists all the papers that that sort of agrees with this query. So this is one example. So moving on, the next level of integration is adding more information, that is through information extraction, adding more, providing more data for the user to sort of address big data queries. And this particularly, this work has been initiated under the ELIXIR Accelerate project a couple of years ago. And what we have done is, under this project, we have established an infrastructure, mainly looking at sharing text mining and data mining outputs on top of the current data that we host in Europe PMC. So what does this infrastructure look like? So this is the main infrastructure of the Europe PMC Annotations platform, which also it involves contributions from other ELIXIR partners. So mainly, the infrastructure supports text mining data or curated data from any source to be included via a submission system. So if a particular text mining group is interested in sharing their data, they have to contact Europe PMC, and they'll be given an account where they can submit their data. And this data is integrated into the annotations database, which then exposes this annotations via an API. And Europe PMC uses this API to highlight those annotations on the original article. Let's see how it works. And also, apart from that, what we do is we have annotation types, different annotation types, covering entities like organism, diseases, protein interactions, gene disease associations, accession numbers, database accession numbers for over 50 databases, and so on and so forth. And this integrating this data is via the W3C recommended web annotation data model that allows us to mix and match text mining annotations coming from different sources. Currently, there are over 10 providers, and they periodically submit data via the submission system, and there are no restrictions in terms of the frequency of the submissions. There are some providers that have provided a one-off dump, and there are providers who submit data on a monthly basis, and Europe PMC contributes to this platform on a daily basis, for instance. So how can, coming back to my original aspect of how you can access this annotations. So there is a typical article page on Europe PMC. As you can see in the right side, you have a View Key Terms tab. Clicking on this tab allows you to view all annotations that have been made on this particular article, coming from different sources. and so it basically lists different types of annotations coming from different sources, and by highlighting them, you're able to mix and match these annotations. And the main goal is to allow users to quickly skim read the article in order for the user to understand what sort of information is described in a particular article. And the other way to access these annotations are by the Annotations API. So, like the Articles API hosted by Europe PMC, the Annotations API also provides different modules to access specifically annotations hosted by the platform. So this can be done in different ways. That is, you can query the API and query in terms of fetching annotations for a set of articles, or you can fetch annotations by specific entity like P53. Then it will fetch all articles that mentions P53. And by provider, if you're interested in a specific provider only, you may choose to fetch annotations by provider. And by relation, and also it allows you to fetch annotation by relationships. That is, currently we serve gene-disease relationship and gene and protein target relationships. And apart from this, you can also search the database by section or type. That is, if you want to query chemicals, you want to fetch all chemical annotations made in mentioned in the method section, you could do that. And there are different formats. The annotations are provided in different formats. That is JSON, JSON-LD, XML, or if you're just interested in the list of article IDs, you can also fetch just the ID list. So let's look at a typical response. So this would correspond to fetching annotation by article ID. So here, the structure of the response is fairly straightforward. You have a section that provides you the article ID, that is both PMC ID and when available, and the abstract ID, PMID. And it provides the specific annotation and the database it is grounding to. And it provides information on which section of the article is coming from, who the provider is, and what type of annotation it is. And also, you can retrieve the annotation URI. I will get back to the annotation URI in a bit. Similarly, you can also, as I mentioned, you can also use annotations by section or type to fetch specific aspects of the database. And in this, accession numbers is something that we have worked on specifically for the ELIXIR data platform. And what the users could do is to fetch all annotations of the type accession numbers of a specific database of interest. For example, here you can see that you're fetching accession numbers from intact. So by selecting this, you can simply fetch all the articles that mentions the accession numbers in the paper. I would like to know that this is added value. That is, this is not necessarily recorded in the databases, the corresponding databases themselves. These are accession number mentioned by the authors in the paper. So by providing this adds another layer of information that was not that could be useful in data analysis. Apart from this, what we had worked on was further deep linking of annotation. So you may have noticed that in a given database, often for a data entry, there are publications that are being cited corresponding to a particular data record, and often these citations are higher level. They are mere mentions of the article from where the curator has extracted the information. For example, in this here you can see the data record for insulin from Nextprod database, and you can see that the curator has provided a specific citation, but the users will not be able to go to the specific sentence from where this citation has been qualified. Where did the curator find where, as in which section of the article and the sentence, the curator found a specific information that has been that has sort of qualified this paper to be associated with this data record. So what we have done is we have worked with Nextprod database and also the group of Patrick Rouge, who was a collaborator within the ELIXIR data platform, to have a pilot work for deep linking of annotations. So Patrick's group submit gene functional annotations to the platform, and correspondingly Nextprod for their data records cite these gene riff annotations in their data. So we wanted to demonstrate that there could be more deeper linking of these annotations from the database to the source text in the article. So, for example, this sort of presents a sample gene functional annotation, and while clicking on the highlighted term, you can see a pop-up, and it gives you a link to the corresponding database. In this case, it is Nextprod. So, on clicking on this, it takes you to the corresponding page in Nextprod database. Alternatively, a user who is visiting the Nextprod database might want to know from where exactly this particular annotation comes from. So, in the link provided, here you can see that there is a mention of the gene riff annotation corresponding to this data record. If the user clicks on this, it will directly go to the specific sentence where the annotation was taken from. So this way, we sort of complete the bidirectional linking of annotations that sort of binds the data and the literature together. And let's see how it looks under the hood. So essentially, there are two aspects. The URI of the annotation is being consumed by Europe PMC for highlighting the annotation on the text, whereas the Nextprod developers use the annotation URI to include in their data record so that it can be linked to the specific text. And we have implemented this mechanism across all annotations. So any developer or anybody who wants to use these annotations and wants to sort of show the veracity of a specific annotation, they can use this URI in order to link to the specific sentence. So this sort of wraps up the work that we have done with respect to literature data linking in the ELIXIR data platform. And I would like to thank all the participants for for listening to this lecture. And I'd also like to acknowledge the contributions made by the various leads of the data platform and the team members of Europe PMC in different capacities, and also the contributors, the collaborators of the Europe PMC Annotation. If you would like to know more about how to search, how to use Europe PMC APIs, we would suggest that you take a look at this specific webinar that we gave a few months back. This describes how to use, it sort of describes all the functionalities in more detail, the API functionality in more detail, and also provides use cases, bioinformatics use cases as to how to use these APIs. And if you would like to have any further questions, please don't hesitate to contact me. I have the email address, or if you have any other questions regarding the annotations, or in general regarding Europe PMC or ELIXIR, the work that Europe PMC does for ELIXIR, please do contact us. Thanks a lot.